today's pre-class exercise, we're going to look at the structure of a eukaryotic cell and its organelles. Organelles are the membrane-enclosed compartments that you find inside of eukaryotic cells. So by the end of today's pre-class exercise, I'd like you to be able to, one, draw a eukaryotic cell and all of its organelles, as well as the features that we'll talk about today, and describe their function. Keep in mind when we do our tour of the cell, this is our animal cell, that this is really a generic cell that is displaying all of the possible organelles in a eukaryotic cell. But in multicellular organisms like ourselves, cells found in different tissues have very specialized functions, and their structure reflects those different functions. And so we find different densities or different amounts of these different organelles and structures. Okay. So I recommend as we walk through the structure of the cell, you might want to look at this diagram that's in your textbook to follow along. Okay. Let's start with what's probably the most defining feature of a cell, and that's the presence of the plasma membrane. The plasma membrane is essential for separating the chemistry and the chemical reactions that are occurring inside the cell from its environment. So it's important for maintaining the cell environment. Okay. Inside of the cell, everything that's inside of the plasma membrane but outside of the organelles is referred to there as the cytosol. So that's the compartment inside of the cell membrane. And in that compartment, in the cytosol, we find cytoplasm. Now, cytoplasm is the material or the substance inside of the cytosol. It's that watery stuff that also would contain ions and proteins. Okay, let's move on next to look at the nucleus, shown here. The nucleus is where our hereditary information is stored in the form of DNA. And the DNA in a nucleus is packaged up in chromosomes. These are structures that are composed of both DNA and protein. Typically, in a cell, you can't see the chromosomes because they are these very thin, thread-like structure. Only as cells are getting ready to divide and those chromosomes become condensed can we actually visualize the chromosomes. But you do see this very electron. I'm going to move my arrow here so that the nucleus is pointing to here, because this dense yellowy orange region here that you can typically see when you used a high-powered microscope called an electron microscope is called the nucleolus. Okay. And it's a dense region that's visible because it contains both RNA and protein. And this is where ribosomes are synthesized. Okay, I'd like to look at the structure of the nucleus in a little bit more detail. The nucleus is one of the three organelles in eukaryotic cells that is enclosed by two membranes. So I'll represent a single phospholipid bilayer as one line. Again, the nucleus is enclosed by two phospholipid bilayers. And studding in this whole structure is referred to as the nuclear envelope. And the nuclear envelope is studded with pores called nuclear pores. These are literally holes in the nuclear envelope that are lined with proteins that allow molecules to move into the nucleus and out of the nucleus. Okay. Continuous with the outer nuclear envelope is the endoplasmic reticulum, shown here. Let me draw it on my diagram here. So, as I said, it's continuous with the outer membrane. And it looks something like this. I'm going to draw a small endoplasmic reticulum. Okay. There are two types of endoplasmic reticulum. There's rough endoplasmic reticulum and smooth endoplasmic reticulum. You can see in the diagram that closer to the nucleus, this region of the ER here, is studded with these yellow balls, and those are ribosomes. So the rough ER is rough because it has these ribosomes associated with the surface of the membrane. I think of it sort of like sandpaper and that grit that's on the surface of the sandpaper. And so the presence of those ribosomes roughs up or makes it, or accounts for what's called the rough ER. So we have rough ER. Ribosomes are the site of protein synthesis. So the rough ER is an important site of protein synthesis. Out here, continuous with the rough endoplasmic reticulum, is the second type of ER, which is called smooth ER. And it's smooth because it doesn't have any ribosomes associated with the surface of the membrane. Rather, the smooth ER is an important site of lipid synthesis. This is where phospholipids are made, and it's also an important site for the storage of calcium. Calcium is an ion that's important for regulating many different reactions in the cell. Okay. So as I said, the rough ER has ribosomes associated with it, shown here. But you notice, there are also ribosomes that are free out in the cytosol. There's nothing different about the structure of ribosomes that are associated with the rough ER or those that are free out in the cytosol. The only difference is that they're making different proteins, and those proteins are going to end up in different locations in the cell. Okay. The next structure I'd like to talk about, or the next organelle, is the Golgi. Okay. The Golgi has this lovely sort of pancake-like structure, which I'm going to draw out here. It's this lovely series of stacks, and those stacks are called cisternae. Okay. The Golgi is an important site of protein processing. For example, this is where carbohydrates may be added to proteins. There's a sidedness to the Golgi. If you notice the stack that's closest to the nucleus, let me look at my diagram here, right here, okay, that pancake in the stack is actually bending towards the nucleus. That cisternae, or that region of the Golgi is referred to as the cis face. And the pancake that's on this side of the stack, closest to the plasma membrane, which is shown right here, is referred to as the transphase. Proteins will move through the stacks of the Golgi and obtain different modifications as they move through the stacks, or the cisternae. And they move from the cis phase of the Golgi to the transphase. Now let's look at organelles that are involved in breaking down things in the cell. First, let's look at the lysosome. The lysosome is the recycling center in the cell. It contains enzymes called acid hydrolases that break down the major biological macromolecules into their monomers, and then those monomers can be reused by the cell. So for example, it breaks down nucleic acids into nucleotides, breaks down proteins into individual amino acids. They're called acid hydrolases because they function in the acidic environment of the lysosomes. The lysosomes have a higher concentration of protons compared to the cytosol. In fact, the pH of the lysosome is only 5, but those acid hydrolases function really well in that low pH or high acidity environment. If they were ever released into the cytosol, where the pH is about 7.2, they wouldn't function properly, and all of your macromolecules would remain intact. The second organelle that breaks things down in the cell is called the peroxisome, shown here. Notice that both of these organelles look similar. They're both spherical organelles that are enclosed by a single membrane, but they have different functions. The peroxisomes are involved in redox reactions, and we'll talk a lot more about those in class. Okay. And some of those redox reactions that are involved in breaking down molecules generate peroxide. Okay. And that's why this organelle is referred to as the peroxisome. Now, peroxide can be very toxic to cells. It damages molecules, but fortunately peroxisomes also have enzymes that break down and get rid of the peroxisome, get rid of the peroxide. Another important thing that's broken down in peroxisomes are fatty acids. And some things that we don't want are also broken down in peroxisomes. For example, in your liver, peroxisomes break down the ethanol that is part of alcoholic beverages that you might enjoy occasionally. Okay. Next, we'll take a visit to my favorite organelle, the mitochondrion, or mitochondria is plural. The mitochondria is often referred to as the powerhouse of the cell because this is where the majority of ATP is produced in the process of oxidative phosphorylation. So it produces ATP. I'm going to abbreviate it as oxidative as OXPHOS for oxidative phosphorylation. Now, the mitochondrion is the second organelle that's enclosed by two membranes rather than one. Let me show you in a little bit more detail down here. So there's an outer membrane, and then there's an inner membrane, also called the cristae, which has lots of surface area. It's highly invaginated, and that's where the enzymes that carry out oxidative phosphorylation are localized. Now let's look at some structures in the cell. These aren't organelles because they're not enclosed in a plasma membrane, but rather are composed of proteins. So the first are these blue lines shown here, representing the cytoskeleton. Just like our own bony skeleton, the cytoskeleton of a cell provides support. But another really cool function of the cytoskeleton is that it drives cell movement. So it moves things around in the cell. Sometimes, for example, the cell wants to move whoop, whoop, the mitochondrion around, whoop, maybe not outside the cell, inside the cell. May want to move an organelle around, may need to move a vesicle, a membrane enclosed compartment around in the cell, and it does that using the cytoskeleton. But the cytoskeleton is also important in some cases for the whole cell to move. So for example, your white blood cells moving to a site of infection involves the cytoskeleton. The other structure in an animal cell, shown here, is the centriole. Again, it's not an organelle because it's not enclosed in a membrane. It's composed of protein, and in fact, it's composed of proteins that also make up the cytoskeleton. It has this very distinctive two barrel-like structures in which the barrels are positioned 90 degrees to one another. It's involved in cell division. Specifically, it's involved in separating chromosomes during cell division. So we'll talk in a lot more detail about the structure and how the centriole works when we talk about the processes of mitosis and meiosis. Okay, now let's turn and look at some of the organelles and structures that are found in plant cells, but not animal cells. So here we have our generic plant cell. First thing I want you to notice is that you don't see centrioles in this plant cell, nor do you see lysosomes. Those are features that we only find in animal cells. Now let's look at what you find in plant cells, but not animal cells. First, you see a cell wall. Okay. The cell wall is important for structure, okay, and support. Okay. Notice that even though it has a cell wall, it also has a plasma membrane inside of that cell wall. Another feature we find in plant cells, but not animal cells, are chloroplasts. And this is where that very important process of photosynthesis occurs, in which carbon dioxide is fixed into sugars. So, sugars are synthesized in photosynthesis. So the chloroplast is the third organelle that's characterized by two membranes. Let me show you. I'm going to draw it up here. Okay, there's an outer membrane and an inner membrane. And the chloroplast also has structures inside called thylakoids. So these are membrane enclosed compartments that are stacked up. Each of these is called a thylakoid, and the entire stack is referred to as a granum, or the plural is grana. Okay? And we'll look at more detail at the structure of the chloroplast and what's happening in each of these compartments when we discuss photosynthesis. The other organelle that you find in plants, but not animals, is a vacuole. And the vacuoles are a site of storage. Water is stored in vacuoles. So when you water your houseplants, you take care of them. That vacuole will increase in size as it fills up with water, and the membrane of the vacuole will push up against the plasma membrane in the cell wall, making your plant stand up nice and tall. And as you neglect your plant and it becomes dehydrated, the vacuole begins to shrink, and your plants, like mine, begin to wither. The other things that you might find stored in a vacuole are chemicals, like caffeine, and also toxins that make plants toxic to some animals. And you also find pigments stored in vacuoles. So this is what makes your flowers nice and colorful. Okay, so that wraps up our tour of the eukaryotic cell. In class, we'll look in more detail at the structure and the function of specialized cells.